Hello, BISC1323. This is the beginning of recorded lecture 2-1. Starting off with the topics for exam number 2 with the chapter on fungi. So let's start by looking at just some of the characteristics, some of the traits that we can associate with fungi. Fungi have a cell wall, but importantly, not peptidoglycan, that was a bacteria thing. Fungi have a cell wall made of a polysaccharide called chitin. And there's other stuff that's part of the cell wall, but the important thing is this polysaccharide chitin, just connected sugars, classic polysaccharide. But yeah, this chitin is associated with fungal cell wall. Some fungi are multicellular, obviously the ones that you buy and eat in a grocery store, but there are also single-celled fungi. There's a term for them, yeasts. It's not a clade, it's just sort of an umbrella term for unicellular fungi, broadly referred to as yeast, but it is an important term that comes up a lot. Fungi that are multicellular are much more interesting in my opinion. They produce these structures called hyphae. There are two types, and let's read the definition of hyphae while we look at these things. The key terms define hyphae as a fungal filament composed of one or more cells. So yeah, this is a thin, it's only one cell layer thick or one cell thick, so these are pretty thin structures. So they're long filaments. This is one that's made of several different cells. You can see each one has its own nuclei. This is called septate hyphae. You'll notice that even though these are separate cells, there's a cell wall that is separating this cell from this cell, from this cell, from this cell. These septa, these walls, have a big pore in the middle. And so these are distinct cells, but the cytoplasm and the nutrients can flow freely from cell to cell very quickly. The other type of hyphae is called cenocytic. It's spelled a weird one, but it's pronounced cenocytic hyphae, where it's just one big long cell in this filament, and there are multiple nuclei spaced throughout this, but this is all one big cell, this single filament here. So obviously we don't see these in yeasts, we see these in multicellular fungi, these hyphae. To summarize, septate hyphae have walls called septa that incompletely separate the cells. Again, we saw those big pores in the septa, and cenocytic hyphae are one single big multinucleated cell. One hypha by itself is not going to do much, so what fungi do is they have a lot of these things. A bunch of these filaments all together make up what's called mycelium. And again, we can look at this while I read the definition. The key terms defines mycelium as a mass of fungal hyphae. These are familiar looking button mushrooms up here, but underneath the surface of the soil, this is where the real action is taking place. This is the mycelium of the fungus, absorbing mineral nutrients from this decaying, looks like wood chips in here. Speaking of absorbing nutrients, that's the purpose of this mycelium, increasing surface area for uptake of nutrients. They're heterotrophs. They're not doing photosynthesis. They're absorbing organic compounds as saprobes. This was a key term in the previous chapter. We described those oomycetes, but new tests, so I pull up the key terms again. A saprobe is an organism that derives nutrients from decaying organic matter. And many classic mushrooms are dead decaying tree, trunk, or branch, or whatever this is, saprobe. That's not the only lifestyle that fungi can have. They're all heterotrophs, but there are a couple of different ways to be a heterotroph. You could break down dead, decaying organic matter. And as a side note, this is a very important part of the carbon cycle and the nitrogen cycle, microbial respiration, helping break down organic matter, sending it back to the atmosphere, nitrogen cycle, breakdown of nitrogen to return nitrogen back to the air. That's also fungi. Bacteria and fungi are important parts of this. But another way to be a heterotroph is to be parasitic. There are several human pathogens. Here's ringworm. It's called ringworm. It's not really a worm at all, it's a fungal infection. Here are some other human fungal infections. Don't memorize all these, but yeah, we got athlete's foot, jock itch. Some of these can infect the lungs. Not going to go through a laundry list of these, but yeah, there are some important parasites that infect humans and many that infect plants. Again, the textbook has a big section of a bunch of these. I don't want to make you memorize all of them, but just know that some of these parasites are important animal and plant pathogens. There's a third way to be a heterotroph other than being a saprobe or being a parasite, and that's to be a predator. And there actually are some fungi that actively hunt their prey. So here's a fungal filament wrapping itself around a nematode roundworm. We'll talk about nematode roundworms when we get to our chapter on animals and invertebrates. But yeah, it's attacking its prey. It's predatory. So again, all of these nutritional modes, as I'm labeling them, they're all types of heterotrophy. Some other things we can call out for fungi are their oxygen tolerance. So a bunch of key terms here. Fungi can be obligate aerobes, obligate anaerobes, or facultative anaerobes. Now, this seems like a lot, but if we break down each one of these terms word by word, it's actually pretty straightforward. Aerobe means oxygen. Aero refers to air. Aerobics means you're breathing hard and taking in a lot of oxygen. So aerobe means oxygen. Anaerobe means without oxygen. Okay, what does the word obligate mean? Obligate means you have an obligation to live this way. You have to be like this. So obligate aerobe means you have to have oxygen. Obligate anaerobe means you have to not have oxygen. And facultative is kind of a weird one, but it just means you don't have an obligation. Facultative anaerobes mean they can survive either way. Here's a visual of this. So if you were to have a tube set up where there's oxygen at the top where it's exposed to the air and low or basically no oxygen at the bottom of the tube, obligate aerobes would only be able to grow at the top where there's oxygen. Obligate anaerobes would only be able to survive at the bottom where there's low concentrations of oxygen. And facultative can, do either one. You'll find them sort of throughout the tube. They usually prefer oxygen, but they can survive down here as well. If you want more concrete examples, are classic mushrooms that obviously live surrounded by air. These are obligate aerobes. Obligate anaerobes include a lot of these microscopic fungi that have different relationships with ruminants. And facultative anaerobes are things like yeast, like brewer's yeast or baker's yeast. They start fermenting beer when you take away the oxygen, but they can survive with oxygen as well. So you don't need to memorize all these examples. You just need to know the key terms definitions, which I've described. Many fungi can reproduce asexually, and in that, there are several types of asexual reproduction. One of these is called fragmentation, where you have some fungal hyphae, some kind of damage, maybe some gardening trowel rips this up, or maybe some digging animal rips this fungus apart. But this fungal filament here, that's a piece of this previous one, it can grow into a new fungus. So fragmentation is when a new organism grows from a fragment of hyphae. Another way to reproduce asexually is through budding. We only see this in yeasts, we only see this in single-cell fungi, but it's where a part of the cell sort of buds out, grows larger and larger and larger until they're both about the same size, and then it pinches off and you got two cells. Asexual reproduction. And there's a third type called spores, which are released from a structure called a sporangium. So here are some fungal hyphae. You have these filaments, hyphae, but look at the tip of this hypha. It has formed a structure called a sporangium. Its whole job is to make spores and release them. And again, there's no sexual reproduction. There's no mixing with egg and sperm or other genetic stuff like that. It's just asexual reproduction, cloning itself, but it is a way to spread these spores and spread itself around. So fragmentation, budding, and spores, all forms of asexual reproduction. In addition to these asexual forms of reproduction, there are many fungi that are also capable of sexual reproduction. So before we get into this, just a quick review here of a couple of terms back from BISC1323, the terms haploid and diploid. Haploid, just to have a visual to this, refers to, and I'm reading off the key terms here, haploid is a cell, nucleus, or organism containing one set of chromosomes, often abbreviated as lowercase n or sometimes 1n, one copy of each. Diploid is, reading from the key terms, a cell, nucleus, or organism containing two sets of chromosomes, and this is usually put as 2n. We are diploid. We think of ourselves as diploid. All of our cells are diploid, and for us, we only go haploid when it comes to making eggs and sperm. However, with fungi, haploid is actually the norm in most fungi. Look at this mycelium here. All the cells of this mycelium in these hyphae are, by default, haploid cells. And, oh, hey, this is what we just talked about, asexual reproduction, making haploid spores, going back to more haploid mycelium. It all stays haploid. But what we're focusing on now is the sexual reproduction side of things. So if we want to mix some genetics up, we're going to take haploid mycelium from one individual and fuse this with haploid mycelium of a different individual. There isn't male and female like there is with animals, but there are different mating types that exist to ensure that there is some genetic diversity. But the first step here is when these two cells fuse their cytoplasms together. This process of fusing cytoplasms is called plasmogamy. And importantly, after plasmogamy, the two nuclei from these two cells are separate from one another. You have a stage where you have this cell that has two nuclei. So to summarize here, and yeah, I'm breaking this down 1, 2, 3, 4 steps. Two mating types of haploid hyphae fuse their cytoplasms. That process is called plasmogamy, fusing cytoplasms. They form what's called a dikaryotic cell. Again, di means two, karyon means nucleus, dikaryotic means a cell with two nuclei. Now, it doesn't stay this way forever. The next step is to fuse those two haploid nuclei into a single diploid nucleus. This is just 1 + 1 = 2. This process of fusing nuclei is called karyogamy. Again, karyon means nucleus, karyogamy is fusing of nuclei. So nuclei fuse to form a single diploid nucleus, that's karyogamy. They form a diploid cell that we call a zygote. Okay, now it's time to make haploid spores. This is the only point at which it's diploid in this sort of generic fungal life cycle. The way they make haploid spores is the same way that we make haploid eggs or sperm. They're going to go through the process of meiosis. Remember all this? So we don't have to reinvent some brand new way of doing this. Obviously, they're not making eggs and sperm like we are, but it's the same basic process. The zygote forms haploid spores by this process of meiosis. And then these spores are released, and they can grow into new haploid mycelium. Yeah, spores grow into haploid mycelium. Okay, so this is a generic fungal life cycle. As we will see when we get into specific phyla, specific groups of fungi, the defining feature of several of these is how they do this slightly differently. So this is an important baseline that we've established, but we will deviate from this when we go to different groups. Speaking of different groups, that's our next section here, classifying fungi. So fungal phylogeny is, ooh boy, okay, there's a lot of stuff here. It looks pretty complicated, but we've got a simplified version. Here are 5 groups of fungi and, animals over here too. We can break fungal phylogeny down into 5 major. First up among these 5 fungal phyla is this group called Chytridiomycota. these are mostly single-celled fungi, mostly unicellular, but some of them are multicellular, and multicellular fungi have hyphae. They have cenocytic hyphae, so yep, remember that's what this means. No septa, no walls. There's something kind of weird about these single-celled chytridiomycota, though. If you look closely, there's this weird line coming off of the cells. Might look familiar. That's a flagellum. These are the only fungi that have flagella present specifically on their spores. We know what flagella do. They're used for swimming, so it should not be surprising that these swimming fungal spores live in the water. So Chytridiomycota are mostly aquatic, and the most notable member is indeed a parasite, something called Batrachochitrium dendrobatidis. Whew. Just a reminder, I am not a stickler for spelling. If I have a question about this on the exam, I'll spell it out for you. And I'm never going to try to trick you by, misspelling this slightly and that makes it the wrong answer or something dumb like that. But anyway, so why is this so notable? Well, I'm not going to try to do it again. This thing is responsible for a disease that affects amphibians. We'll talk more about amphibians later in the quarter when we get to our chapter on vertebrates, but the short version is they have particularly thin skin, which makes them susceptible to fungal infections. And it's not just killing frogs, it's killing lots of amphibians, having a huge impact on them across the planet. So chytridiomycosis is the name of this disease, and yep, I can say devastating amphibian populations across the globe. That's all I wanted to say about this phylum. Again, not a very major one, but the next one will bring something maybe more familiar to us. Next, I want to talk about Zygomycota. So Zygomycota also have cenocytic hyphae. We remember what that means, but the claim to fame of Zygomycota is a unique life cycle. So, okay, I told you this was coming. This is a slight variation on that sort of generic life cycle that we looked at before. I'm not gonna, go through all of the steps here again. I really just want to point out the part where this deviates from the norm. So we've got asexual reproduction, that's normal. We've got, positive and negative mating types, haploid mycelia, fusing together. But the part that is worth noting is this part down here. This zygote cell is a spore. This diploid zygote does not just immediately go through meiosis to make haploid spores. This diploid cell is itself a type of spore, conveniently called a zygospore, produced by Zygomycota, love it when the name makes sense, that resists harsh conditions and can stay there for a while before it goes through meiosis and makes spores and then releases them and sort of completes the cycle. So in summary, these Zygomycota form what are called zygospores during their sexual reproduction. These are diploid spores, as opposed to the normally haploid spores, diploid spores that resist harsh environments. And when conditions are good, they can germinate, form stalks, and release haploid spores from a sporangium. That's business as usual, but this part is a little bit different. The most notable member, I think, of Zygomycota is something called Rhizopus stolonifer. You would know this as black bread mold. If you ever had a loaf of bread that you left out on the counter for maybe a little too long, it started getting fuzzy like this. If you were to zoom in on this gross-looking black fuzz, you would see these white filaments, and those little black dots, those are sporangia, again being elevated up to try to get caught in the wind and disperse these spores as distantly as possible. So it's called black bread mold because you can see the black dots here. So that's our most famous Zygomycota. Moving on, let's do Ascomycota next. So, yep, we can see one perhaps familiar face here. This is a morel, but what's the deal with Ascomycota? Well, it's another life cycle thing. So, The asexual part of it's the same, haploid mycelia, making haploid spores, growing into haploid mycelium. Okay, that's easy. We've got plasmogamy, where two mating strains will fuse and form this dikaryotic cell, but here's where it goes away from the norm. Usually in our sort of typical generic life cycle, there was just one dikaryotic cell that quickly fused cytoplasms to become diploid and then, went through the rest of this stuff. Instead, in Ascomycota, this dikaryotic cell with two nuclei goes through several rounds of mitotic cell division. So we get many dikaryotic cells. We get this whole structure called an ascocarp, sort of like a cup, called an ascocarp, that is all made of dikaryotic cells. And something special is going to happen at these two cells located at the rim of the cup, the tip of the cup, however you want to describe that. These cells are called the asci or ascus. So let's summarize this before we go on. So in Ascomycota, again, not going through the entire life cycle, just highlighting the part that's a little different. After plasmogamy occurs, a cup-like ascocarp forms that's entirely made of dikaryotic cells. Cells at the rim of this cup are called asci or ascus. It's the plural singular. So at the ascus cell in the ascocarp, we finally get 1 + 1 = 2. We finally get karyogamy to form this diploid zygote. Then we have meiosis, and at this point we're starting to count things. You may remember meiosis from BISC1323, that meiosis takes one cell and through meiosis 1 and 2 ends up with a total of 4 cells, because there are 2 rounds of cell division, meiosis 1 and meiosis 2. But what's happening here is we're having karyogamy to produce a single diploid cell, meiosis to produce 4 haploid cells, and then mitosis cell division. So each one of these 4 cells that comes from meiosis replicates itself again. That goes from 4 to 8. If you're getting lost in the math here, it's a lot simpler than it sounds. The ascus cell is dikaryotic. 1n + 1n is a way to do it. 2 haploid nuclei. Karyogamy is 1 + 1 = 2. We go to a zygote, which is diploid. Then we have meiosis, which goes from 1 diploid cell to 4 haploid cells. Then we have mitotic division, where we go from 4 haploid spores to a total of 8 haploid spores. Again, this is distinctive when looking at these things and trying to identify fungi. Seeing this cluster of exactly, precisely 8 ascospores is a big deal when identifying these Ascomycota. So then finally this, ascus cell does karyogamy, meiosis, then mitosis. We get 8 haploid ascospores. So what about notable members? Well, there are a bunch of notable members. To rattle off some of these, they include Candida albicans, a pathogen responsible for oral thrush. Google that one at your own risk. Yeast infections, morels and truffles, mycorrhizae, lichen, and the 2 that I want to call out in particular, penicillium. This is our source of the antibiotic penicillin. Yeah, we did not invent penicillin. We stole this from nature. These fungi have been waging war against bacteria for longer than we've been around. So this penicillium is our source of penicillin, and it's an Ascomycota, as well as Saccharomyces cerevisiae. This is our baker's yeast. This is our brewer's yeast. This is how we make the bread. This is how we make the beer. These are both. All right. With Ascomycota out of the way, we've only got a couple left. Let's do Basidiomycota next. So the distinguishing feature of Basidiomycota is another another life cycle, another unique way of doing sexual reproduction. So let's take a look at this. We've got haploid mycelium and a different mating type of haploid mycelium. These cells fuse through plasmogamy, so we've got a dikaryotic cell. And similarly to zygomycota, these dikaryotic cells divide. You can see how there are a bunch of dikaryotic cells, but instead of stopping with just, a small cup of dikaryotic cells, these basidiomycota go even further. They form so many dikaryotic cells that it forms its own dikaryotic mycelium. This is kind of like two organisms in one, because this is haploid mycelium growing and absorbing nutrients and doing its thing. This dikaryotic mycelium is a whole nother entity. And in fact, it's the mushrooms that we know and love that are made of entirely dikaryotic mycelium. So yeah, this big, huge multicellular structure is all made of these dikaryotic cells. Now, they can't stay dikaryotic forever. We have to do karyogamy at some point. So in these structures, the technical term for a mushroom is a basidiocarp. Basidiomycota have a basidiocarp. It's nice when the names line up. In these gills of the mushroom are structures called basidia. And it's in the basidia that we do 1 + 1 = 2. We have karyogamy, and a dikaryotic cell becomes a diploid cell. Then it goes through meiosis and releases these basidiospores to grow into more haploid mycelium. There's only 4 here because we're doing karyogamy, then meiosis, no mitosis after the meiosis. So it's just a simple 1 goes to 4, and then these are released to form haploid mycelium. How do we summarize this? So the haploid primary mycelium undergoes plasmogamy and then mitosis a bunch of times to form this big dikaryotic secondary mycelium. The secondary mycelium is the dominant structure, again, the thing that we see most of, including the basidiocarp, the mushroom. And it's within the club-shaped basidia in the gills of this mushroom that we finally have karyogamy, then meiosis, resulting in 4 haploid basidiospores. They're going to be released, grow into primary mycelium, and then the cycle can repeat itself. Notable members of Basidiomycota. Obviously, all of our mushrooms, some of them edible, some of them very not edible, and shelf fungus. There's a lot of these that are big as well. So shelf fungus and all of these toadstool-style mushrooms are members of Basidiomycota. Now, there's one more phylum, but before I get into that, I have a side note. There are some ascomycetes, members of the Ascomycota, and some basidiomycetes that cannot reproduce sexually. So I know that we just said that their whole thing was this unique sexual reproduction cycle for Basidiomycota and for Ascomycota, but there are some members of both of these groups that just don't do any of this at all. Some Ascomycetes and some Basidiomycetes that just can't do sexual reproduction at all. These are called imperfect fungi or mold. These used to be classified as their own thing because, as you can imagine, this thing that doesn't do sexual reproduction at all, why would you put this with Basidiomycota, which their whole thing is this unique sexual reproduction? Well, it's only through, stop me if you've heard this one before, modern genome sequencing and sequence alignment technology that we know these weird molds and imperfect fungi, even though they don't behave at all like the Ascomycetes and Basidiomycetes, they are members of these phyla that have lost this ability to do sexual reproduction and instead only reproduce asexually. So when you hear the term imperfect fungi or mold, it's specifically referring to ascomycetes and basidiomycetes that don't do sexual reproduction. And again, this is not a clade. These are just fungi of these two groups that don't do sexual reproduction. I'm putting such a fine point on that because our final phylum, Glomeromycota, their whole deal is they're only asexual reproduction. They don't do sexual reproduction at all. These are not imperfect fungi. These are not molds. Again, this is no sexual reproduction, but a member of one of these two groups. This is a separate group that also is only capable of doing asexual reproduction. And notable members, notable members include mycorrhizal fungi. What are mycorrhizal fungi? Well, we're going to learn about them next time. So this is, we're not done with fungi yet, but this is where I'm going to end things with lecture 2.1. We'll pick back up and learn more about mycorrhizal fungi and more fungi stuff in the next one. This is the end of recorded lecture 2.1.